Hi everyone, and welcome to the Chapter 12 lecture for Chemistry 100. This chapter is all about metabolism and really catabolism, how we take all those macronutrients that we just talked about, carbohydrates, fats, and proteins, and convert them into energy for our cells. So we're going to give an overview of the steps of metabolism, or what metabolism is, the processes, and then we'll be introduced to some of the key players in metabolism, and then we'll talk about each of the sort of steps of the key metabolic pathways in our cells. We'll talk about digestion, the breakdown of those macronutrients that occurs in the intestines, and then how glucose in particular gets metabolized into ATP, or energy for the cell, through three steps: glycolysis, the citric acid cycle, and the electron transport chain. And then we'll touch a little bit on how fats and proteins can be converted to ATP. Fats primarily are for energy storage, so that's what this little funny comic is about, how there's pretty much an endless storage of fat in our bodies. All right, so metabolism is defined as the sum of all chemical reactions in the cell. So all of the chemistry that's going on in the cell is referring to our metabolism. When we talk about metabolism, sort of colloquially, we talk about it as if it's a measure of how many calories we're burning. So that would be discussing just the endothermic reactions that are using energy, but there's also exothermic reactions that are producing energy. And so both of those different types of reactions play a role in metabolism. So we can divide metabolism up into sort of two arms. The arms that are breakdown reactions, that is called catabolism. So where we take large biomolecules like starch, for example, and break it down into smaller building blocks like individual glucose molecules, right? So catabolism involves breakdown. Breakdown reactions involve hydrolysis. Hydrolysis reactions are decomposition reactions, and they tend to be exothermic. They release or produce energy, versus building reactions where we take small things like glucose, for example. If we need to store it as glycogen, we need to link it together to build a large molecule. That is part of anabolism, which are synthesis reactions. They involve condensation reactions, the opposite of hydrolysis, and they are endothermic. They require energy. It takes energy to build stuff. So quick sample problem. Identify the following as catabolic or anabolic reactions. A, digestion of polysaccharides. Well, that would be breaking down. Digesting is breaking down, so that would be catabolism. Synthesis of proteins, B. You are synthesizing something, you're building it, that would be anabolic. And C, oxidation of glucose to carbon dioxide and water. Glucose is a larger molecule than carbon dioxide and water. You are breaking it down. That would be a part of a catabolic reaction. So let's start with catabolism, some more detail. An overview of catabolism. This slide is pretty busy, but I'm going to walk you through it step by step throughout the lecture. So the stages of catabolism, for example, when you are eating a burrito or little chocolate brownies, in the case of my daughter right now, the biomolecules in the food get broken down through a process of hydrolysis. So all the proteins and the starch and the lipids in those foods will get broken down into their building block components. So proteins get broken down into amino acids, polysaccharides or carbohydrates get broken down into monosaccharides, and lipids get broken down into glycerol and free fatty acids. Right? So now these products, these hydrolysis products, are small enough to actually be absorbed into the through the intestinal wall into the cells. And once they get absorbed into the cells, they can be converted into common metabolites. So one common one that is, or two common ones, are pyruvate and acetyl-CoA. So acetyl-CoA is the starting material for the next stage of metabolism, the citric acid cycle. And then it puts out some electron carriers that carry electrons to the electron transport chain, which is where a majority of the ATP is made. So we break down large molecules, then those smaller molecules can get absorbed into our cells, converted into metabolites where they're sent through these different metabolic pathways that ultimately churn out ATP. So we need to orient ourselves in the cell as well. So if this is an animal cell, here's the nucleus, here's the endoplasmic reticulum and the Golgi apparatus. So there's lots of different organelles in here, right? But the one that's most important in energy is the mitochondria right here. So they look kind of like kidney beans, usually in pictures. If you cut them open, they have this squiggly second membrane, an internal membrane. And sometimes mitochondria are referred to as the powerhouses of the cell because this is where the citric acid cycle takes place and where the electron transport chain is located in order for energy to be generated. So mitochondria are just ATP factories, basically. So what is ATP? ATP is a molecule, and it's not the only important molecule in metabolism. So we're going to talk about these 4 different molecules. ATP, NADH, FADH2, and acetyl-CoA are 4 important metabolites that we're going to talk about. These are the high energy forms of them. There's also a low energy form of each one. So ATP, when it loses its third phosphate, becomes ADP and no longer has that same energy. NADH and FADH2 are electron carriers, and in their reduced state, when they have gained electrons and hydrogens, they are in the high energy form versus their low energy form, which is the oxidized state. And acetyl-CoA is the high energy form of coenzyme A. So let's look at ATP first. ATP stands for adenosine triphosphate. So it's an adenosine molecule with 3 phosphate groups on it. And when it's hydrolyzed, when it reacts with water, it's a very exothermic reaction. It produces a lot of energy, and that energy can be captured to fuel chemical reactions. And what's left is ADP, adenosine diphosphate, and a free phosphate group. And through the citric acid cycle and the electron transport chain, we basically run this reaction in reverse in order to regenerate our ATP store. So we use it and then we regenerate it, and use it and then regenerate it. NADH and FADH2 are electron carriers. So the high energy form with the hydrogens added are reduced, because reduction is gain of hydrogens, and the low energy form is the oxidized form. So when I say that they are electron carriers, they are basically capturing electrons that are lost in redox reactions, and they transport those electrons to the electron transport chain. So they capture electrons lost and they take them to the electron transport chain. Interesting thing about these 2 molecules is they both contain a vitamin component. So NADH, the N, stands for nicotinamide. Nicotinamide is vitamin B3. It's from niacin, derived from niacin, vitamin B3. And FAD and FADH2 contain the riboflavin component. Riboflavin is vitamin B2. So you hear a lot with B vitamins. People will talk about, oh, B vitamins are in like energy supplements, or B vitamin supplements claim to give you increased energy. And where that's relevant, what it's talking about is that B vitamins do in fact play a role in energy. They play a role in the energy generating pathway of the citric acid cycle and the electron transport chain. However, if you have adequate amounts of those vitamins in your diet, getting more of them doesn't actually give you more energy. That's enzyme kinetics for you. But it does ensure that you're getting the maximum amount of energy that your cells can make. And then the the fourth molecule here is acetyl-CoA versus CoA. So acetyl-CoA also contains a vitamin component. It contains pantothenic acid, which is vitamin B5. So, all right, so the first step of catabolism is digestion. It's how we take large biomolecules and break them up into smaller biomolecules. The first biomolecule we'll talk about is carbohydrates. So carbohydrate digestion. Starch, amylose and amylopectin are our polysaccharides. They're made up of long chains of glucose, and we break them down through enzymes. We digest them ultimately into all monosaccharides by breaking those alpha glycosidic linkages between the glucose molecules. One of the enzymes that does this is called amylase. Amylase is a starch-cutting enzyme. It cuts amylose and amylopectin, so it's got that same prefix of amylo or word root. So starch will slowly get cut up into oligosaccharides and disaccharides, but ultimately it will be completely digested into monosaccharides of glucose. The disaccharides can all be digested by individual enzymes that recognize them. So maltose, which is 2 glucose molecules, is cleaved or cut by the enzyme maltase. Lactose is cut by the enzyme lactase, and sucrose is cut by the enzyme sucrase. So that ultimately we are left with a bunch of monosaccharides at the end of digestion, and these monosaccharides are now small enough to actually be absorbed through the cells into the bloodstream. So these enzymes are made either by the pancreas. Pancreas makes a lot of them, though the jejunum, part of the small intestine, is actually where lactase is produced. People who are lactose intolerant lack this enzyme lactase, so they can cut up and digest maltose and they can digest sucrose, but they can't digest lactose. And so that sugar builds up in their intestines and it attracts water through osmosis, which gives them diarrhea, and it can also be consumed by bacteria in the intestines, which produce gas as a byproduct, which makes you gassy. So the symptoms of lactose intolerance. Just a reminder that as humans and many mammals only have enzymes to cut alpha glycosidic linkages, to break down starch, amylose and amylopectin. We do not have the enzyme that's called cellulase that's required to break down the beta glucose linkages that are found in cellulose. So we still consume it. It comes out looking very much the same on the other end. I still like to tell this story. One of my friends in college, we were out to dinner, and she went to the bathroom, and she came back to the table, and she was like giggling hysterically. We asked, what's so funny? And she said she was in the bathroom. There was someone in the stall next to her, and all of a sudden she hears, "Corn? When did I eat corn?" Because corn comes out in your stool looking basically the same. The corn kernel is just full of cellulose, of insoluble fiber. So next time you eat corn, check your poop. You'll see the corn in it pretty much unchanged. So yeah, we cannot digest insoluble fiber, but it's still actually very healthy for us and nutritious. It helps to stimulate the bacteria in our intestines to produce compounds that are important for our intestinal integrity. It helps provide bulk to the stool so you can push it out, but it does not get digested. In other words, those bonds between the glucose molecules do not get broken. Only the alpha linkages in starch can be broken down. All right, so that is carb digestion. Fat digestion. Fats are a different type of molecule. They are not polymers, so they're not long chains of repeating units like carbohydrates and proteins are. They're made up of glycerol and 3 fatty acids. and they are nonpolar, so they don't mix well with water. And in order to get them to mix better with water, we have to add an emulsifier. So the body's emulsifier in digestion is bile salts. So bile is made in the liver, and then it's stored in the gallbladder, and then you squirt it from the gallbladder into the small intestine when you eat something fatty. And it helps take these giant lipid globs in your stomach, in your digestive juices, and break it up into smaller fat globs that are easier to get at with the lipase enzymes, the enzymes that are going to actually break down your lipids. So remember, emulsifiers can form micelles. They can form those structures where they've got the polar heads on the outside and the nonpolar tails on the inside, and they trap a little bit of fat in each one. And that process is called emulsification. So when fats are digested, the lipase, the enzyme that digests fats, snips off some of those fatty acids. So it snips off 2 fatty acids. So we turn that triglyceride into a monoglyceride, a glycerol with 1 fatty acid, and 2 free fatty acids. We have to cut it up that way so that it can fit through the intestinal lining and be absorbed into the bloodstream. Once it's back into the bloodstream, though, we actually reassemble it into a triglyceride. So triglycerides are too large to absorb. We do have to digest them, but then we reassemble them. But they're still nonpolar, and we can't transport them through the blood very well. So they actually get absorbed into the lymphatic system, into the lacteal. And when we transport them in the blood, we package them in proteins and phospholipid packages that are called lipoproteins. So a lipoprotein is made up of phospholipid, like a phospholipid micelle, with proteins embedded in it. And we call these special lipoproteins that are formed in the intestines, they're called chylomicrons. So chylomicrons are formed from phospholipids and protein, and they carry our cholesterol and our fatty acids and our triglycerides through the blood in this package that is soluble in the blood. And then lastly, we've got protein digestion. So proteins are polymers like carbohydrates are. So they're made up of a bunch of amino acids linked together. And so the enzymes that break down proteins are called proteases. Ase is a suffix that just means a protein, or sorry, an enzyme. And so if this is our amino acid chain, all right, the first thing we have to do before we can really start digesting the protein is we need to denature the protein. So remember, proteins are folded, they have a 3-dimensional shape, and when you unfold them, it's easier for the enzymes, the proteases, to get in there and do their cut. So denaturing does not involve breaking peptide bonds. It just involves breaking attractive forces between side chains that hold the 3-dimensional shape together. So once it is denatured, either by heat or by stomach acid, well, really, if you're cooking the food, you might have denatured the protein already with heat. In your stomach, the stomach acid helps to denature the proteins. And then we can start going at it with our cutting enzymes, the proteases like pepsin and trypsin and chymotrypsin are all proteases that digest or break peptide bonds. Once we have them down to single amino acids or even dipeptides and tripeptides are small enough to be absorbed into the intestines and into the through the intestines into the bloodstream. So we have taken carbohydrates, fats, and proteins and broken them down into smaller components, absorbed them into the bloodstream. Quick enzyme question. Indicate the enzymes that are involved in the digestion of the following. Lactose, that is involved in digesting, or the enzyme that digests lactose, a disaccharide sugar, is lactase. Lactase is the enzyme we're looking for. what enzyme is involved in digesting triglycerides? That's the enzyme that breaks down lipids. It's called lipase. And some that break down protein as a class, they're known as proteases, but some specific ones to know are pepsin, which is in the stomach, and trypsin and chymotrypsin, which are in the small intestine. I do recommend that you make a list and know all these different enzymes, where they're located and what they act on, carbs, fats, or proteins. Okay, so now that we have everything down to these basic building blocks, we have the carbs digested into monosaccharides, and the proteins are amino acids, and the lipids have been broken down into fatty acids, okay? We're going to follow the carbohydrates through the energy producing process. So the first step of that metabolic pathway is glycolysis. Maybe the first metabolic pathway is glycolysis. So glycolysis takes place in the cytosol of cells. So glucose gets taken up into the bloodstream, and it goes through the bloodstream, and it becomes available for different cells to take up. So a cell can take in glucose through the glucose transporter in its cell membrane. And then the glucose is inside the cell, it's in the cytoplasm, that jelly part of the cell. And this is where glycolysis occurs. So glycolysis is a 10-step process that converts glucose into pyruvate. One glucose gets converted into 2 pyruvates, right? There's some other molecules that are involved in this reaction, but that's sort of the key feature of it. So we have this written as one reaction arrow, but really think of this as 10 separate reaction arrows. There's 10 separate steps that go on in order for glucose to be converted into 2 pyruvate molecules. So glucose is a 6-carbon molecule. Pyruvate is a 3-carbon molecule. You're kind of just cutting it in half. You'll also notice that one of the products of glycolysis is 2 ATPs. So we do make a little bit of ATP with glycolysis, but 2 is not a lot. You'll see most of our ATP is going to be generated later on at the electron transport chain. Another thing that's generated in glycolysis is 2 NADHs. Remember, NADH is like an electron carrier. It's like a little molecular taxi that taxis electrons from one reaction to another. So glycolysis can be broken down into sort of 2 phases. I can't seem to move my head out of the way. So we'll start with phase 1, the first half, the first 5 steps of glycolysis are known as the energy investing stage, because you actually have to spend ATP in this. So these are reactions that are numbered. So the first reaction, second, third, fourth, fifth. The 5th step is when we take that 6-carbon molecule and split it into 2 3-carbon molecules. Right, but the first and third steps require an ATP energy investment. So we actually use 2 ATPs in that first step, the first half of glycolysis. But in the second half of glycolysis, we generate 4 ATPs. So 4 minus 2 is still a net gain of 2 ATPs in the end. So that's why glycolysis does actually generate a little bit of ATP. You'll also notice here, step 6, this is where that NADH is made, where NAD plus is reduced, so it gains hydrogen and becomes NADH. I also have a star here in the first half next to step 3. We'll see that this is the regulatory step of glycolysis. So a lot of metabolic pathways have regulatory steps. It's a step that basically has an on-off switch. And if you want to stop doing that metabolic pathway, you turn it off, or you can start it by turning it on at that step. And so step 3 here is our regulatory step. So what do we mean by that? So if you have a lot of glucose and you are breaking it down and you're making plenty of ATP and the cell has plenty of ATP, you don't need to continue doing glycolysis. That is a waste of energy. Our cells do not have a mechanism for storing ATP. We make it and then we need to use it relatively quickly. So we can't stockpile ATP. So once we have a certain amount in our cells, we stop making it and we can store the glucose, or we can store the triglycerides as fat, and then we can start doing glycolysis when we are ready for more ATP. And so ATP actually serves as an inhibitor of this enzyme, phosphofructokinase, or PFK for short, which is the enzyme that catalyzes step 3 of glycolysis. So when you get a buildup of ATP, it actually blocks phosphofructokinase and stops glycolysis from continuing. And when the ATP levels in the cell go down, ATP is removed from phosphofructokinase and glycolysis continues. This is called a negative feedback loop, where the product of a metabolic process feeds back and negates it or inhibits it. So glucose metabolism and fructose metabolism are slightly different. And you hear a lot in the media about how, oh, fructose is so bad. High fructose corn syrup is so bad for us. And there is some validity to that. So fructose is regulated differently. In fact, it's not regulated the way glucose is. So in both of these molecules, so here is the metabolism of fructose, the first few steps of fructose metabolism that ultimately result in glyceride, or G3P, and now I can't remember what the abbreviation is for dihydroxyacetone phosphate, DHP. And so you can see in glucose metabolism also at step 5, that's where we get those two molecules formed, okay? So at this step here, these two processes continue the same, all right, from that point forward. But fructose doesn't have a regulatory step, all right? Glucose metabolism has that regulatory step right here where phosphofructokinase is, so we can stop doing glycolysis when we have enough ATP, but we don't stop doing whatever the glycolysis of fructose is called, okay? And so then we end up with a buildup of pyruvate at the end of fructose metabolism, and it ultimately ends up getting stored as fat. So that's why people make the claim that fructose is more fattening than glucose, because we don't have a way to stop breaking it down. We end up just storing it as fat. We end up converting it to pyruvate and storing it as fat. But the main issue with sugar being fattening is just getting too much of it. If you're getting the right amount of calories from fructose and or glucose, you're not going to store any extra as fat. So really it's the quantity, not the quality of the sugar that counts in the long run. All right, so we said that glycolysis converts glucose into pyruvate, all right? So glucose, 6-carbon molecule, gets converted into pyruvate or pyruvic acid. And the pyruvate has two different possible fates. If oxygen is around, which usually it is, it will undergo aerobic metabolism. Aerobic means in the presence of oxygen. So aerobic metabolism is where it goes to the electron transport chain. It goes through the citric acid cycle. I guess I should say the citric acid cycle is aerobic metabolism. The citric acid cycle and the electron transport chain. So you'll see that we generate carbon dioxide in the citric acid cycle and water at the end of the electron transport chain. So those are byproducts of aerobic metabolism. We also generate a bunch of ATP, 36 to 38 ATPs for each glucose molecule. If there's no oxygen around, then we call that anaerobic conditions. And anaerobic metabolism is a different pathway where the pyruvate instead gets converted into lactic acid, or in some organisms can be converted into alcohol. But since we're trying to kind of stick with human cells in this class, our human cells will convert can convert glucose to lactic acid. When would our cells be in a condition where there is no oxygen? Well, a good example is in the muscles when you're exercising. Because when you're exercising, your muscles are going through oxygen so quickly, they're using up oxygen faster than they're using up the glucose. And so some of the glucose ends up going through a separate pathway, an anaerobic pathway, when there's low oxygen in the muscles. So these two different things that can happen. If oxygen is around, if it's aerobic conditions, then pyruvate gets converted into acetyl-CoA. It gets oxidized. So this coenzyme A gets oxidized by the pyruvate, and we end up with acetyl-CoA. If there's no oxygen around, then pyruvate gets reduced. It gains hydrogens, and it becomes lactate or lactic acid. And lactic acid is thought to be the cause of that sort of stinging, burning sensation in the muscles when you're exercising very hard. It is a myth that lactic acid builds up in the muscles and causes muscle soreness the next day. That is not true. Muscle soreness is caused by microscopic tears to the muscle tissue after exercise that then have to repair themselves. All right, so far we talked about glycolysis happening in the cytosol, the cytoplasm of the cell. So we take glucose up through the glucose transporter into the cytoplasm, and glycolysis occurs right there in the cytoplasm. All right, so glucose gets converted to pyruvate. If oxygen is around, that pyruvate will get transferred to the mitochondrion, where it gets converted into acetyl-CoA and gets fed into that TCA cycle, or the citric acid cycle, and the electron transport chain. If there is no oxygen around, though, the pyruvate will get converted to lactate right there in the cytoplasm, and that has its own sort of metabolic pathway that does produce a little bit of ATP, but not nearly as efficiently as as aerobic metabolism. So let's look at that process of aerobic metabolism. And that cycle, this cycle right here, it's called the TCA cycle on this slide, but it has many names. So the citric acid cycle is the one that this textbook uses the most, but other textbooks you might see it listed as the TCA cycle, which stands for tricarboxylic acid cycle, or the Krebs cycle, which is named after a couple of chemists who first identified and described this cycle. So we take glucose in glycolysis, we convert it to pyruvate, and then under aerobic conditions, we convert the pyruvate into acetyl-CoA. So for one glucose, for each glucose, we're actually getting 2 acetyl-CoAs, and acetyl-CoA is what feeds into the citric acid cycle. So for every 1 glucose, we actually get 2 turns of the citric acid cycle. So that's a little math to remember. All right, so the citric acid cycle, of course, also known as the TCA and also known as the Krebs cycle. I tried to get different images from different textbooks that used each of these different terms for it. Biology textbooks tend to call it the Krebs cycle, and chemistry textbooks tend to call it TCA, or the citric acid cycle. So there are 8 steps in the citric acid cycle. You don't have to know them all. We're not going to talk about them. We're going to talk about the starting material. So you should know that acetyl-CoA is sort of the starting material for the citric acid cycle, and that the important products of the citric acid cycle are carbon dioxide, right? That's where, when we talk about carbon dioxide being a waste product of metabolism, it is a waste product of the TCA cycle. So carbon dioxide then builds up and we have to exhale it. Another product is NADH and FADH2, some more of these electron carriers that capture electrons like little taxis and are going to drive them to the electron transport chain at the end of this whole path. You also get a GTP generated. GTP is basically equivalent to an ATP, so it does form a little bit of ATP as well. So And this happens in the mitochondrial matrix, which is the inner jelly of the mitochondria. So far, let's do a little tally of how much ATP we've generated. So we did glycolysis. Glycolysis is 10 steps to convert glucose to pyruvate, and it produces 2 NADHs, which are not ATPs, and it produces 2 ATPs. Okay, so we've got 2 ATPs. And then we do, we're assuming it's aerobic metabolism, so we oxidize 2 pyruvates into acetyl-CoAs. We don't make any ATP in that step. And then the citric acid cycle, we do 2 loops of that, and we only generate 2 ATPs. So that's only 4 ATPs that we've made, but we've made all this NADH and some FADH too. So what do they do? Right? They carry electrons to the last part, the electron transport chain. So they drop off these electrons. The electron transport chain, by the way, is located, it's a protein complex, a complex of proteins that are located in the inner membrane of the mitochondria. And you're going to see that electrons sort of flow through it, generating a sort of electricity that pumps protons into this intermembrane space here, the space between the inner membrane and the outer membrane of the mitochondria. And at the end of that electron chain, the protons come back in through the last complex, and we generate ATP through a process called oxidative phosphorylation. So this is an image showing you a mitochondria, and if we take a little zoom in, you can see the outer membrane phospholipid bilayer and the inner membrane phospholipid bilayer. So here are the different protein complexes of the electron transport chain. There's 5 different ones, complex 1, 2, 3, 4, and I don't know why complex 5 is written over here. Complex 5 is also called ATP synthase. It's the one that synthesizes or makes ATP. There's also 2 mobile carriers. So there's coenzyme Q and cytochrome C, which are also part of the electron transport chain. So this image from your textbook I really like. It's very comprehensive, and this is a good visual of what's happening in the electron transport chain. Okay, so first we have our electron carriers, NADH and FADH2. Remember, these guys are taxiing electrons. So we see them here dropping off their electrons at one of these purple complexes. So NADH drops off electrons at complex 1, and FADH2 drops off electrons at complex 2. The red arrows show the flow of electrons. All right, flow of electrons by definition is electricity. That's what electricity is. And electricity fuels things, right? So in this case, what electricity is actually fueling, the electron flow is fueling these pumps. Complex 1, complex 3, and complex 4 are pumps, and what they are pumping are protons, hydrogen ions. So they pump these protons into this intermembrane space, and you end up with a whole bunch of positive charge in the intermembrane space, and that leaves a negative charge or a charge deficit on the inner membrane in the matrix. And so this is called an electrochemical gradient. So you're setting up this difference in charge, but also in chemicals, because the hydrogen is now concentrated on this side of the membrane. And when you have a high concentration of any substance, it wants to diffuse, right? It wants to move down its concentration gradient. That's passive. And it can do so. So all of these other parts of the membrane block hydrogen ions from moving through. The only place they can go through that they can diffuse down their concentration gradient is in this last complex here, complex 5, all right, which allows protons in. And it actually has a sort of rotor in it that spins. And that flow, that natural flow of hydrogens down their concentration gradient actually powers this rotor, all right? The hydrogens when they come in, they pair up with oxygen, which is the final electron receptor. So oxygen binds to hydrogens, the hydrogen ions that come through, and also the electrons that pass out at the other end of the electron transport chain. And so we combine them to make water, H2O. This process here is very exothermic. It gives off a lot of energy. And we use that energy to put a phosphate group on an ADP molecule to regenerate ATP. So this reaction here, I'll even write this out, all right, is very exothermic. It generates energy. And what is this reaction here? It's oxidation. We're oxidizing these hydrogens, and we are generating energy that is then going to go to fuel this reaction here, this phosphorylation reaction, which is very endothermic. So this is a very common theme in biochemistry. It's called coupling, reaction coupling, where one reaction fuels another reaction. And so this process of making ATP is known as oxidative phosphorylation because it involves the coupling of this oxidation reaction and this phosphorylation reaction over here. So if you don't have oxygen, if you're not breathing, all right, and you're not getting oxygen to your cells, all right, then you can't do this oxidation reaction. You won't have oxygen to do this oxidation reaction, which means your cells won't be able to generate the phosphorylation reaction and make ATP. This is why oxygen is so vitally important to us, because we need it to make energy. We can't make energy without it because we need the oxygen to be this final electron acceptor so we can do this exothermic reaction and fuel this endothermic reaction. So that's what oxygen's all about. This right here is exactly why we breathe. All right, so this is just a summary of that previous slide. What's going on in that slide. NADH and FADH2 are our electron taxis. They drive the electrons from the citric acid cycle to the electron transport chain, where they drop them off at either complex 1 or complex 3. And the electrons flow through these complexes, and as they're flowing through a series of redox reactions, actually, they fuel the pumping of protons into the intermembrane space, protons or hydrogen ions, whichever you want to call it. And that builds up an electrochemical gradient, a distribution of charge and concentration differences on either side of that membrane. And then when those protons move back into the matrix through that final protein complex 5, the ATP synthase, that coupling of the oxidation reaction fuels the phosphorylation reaction. So if we were to uncouple those reactions, though, when the hydrogen moves back into, we can uncouple those reactions by preventing that buildup of that gradient. So an uncoupler is something that, like a protein channel that goes into the membrane and allows hydrogens to flow freely back through. So they don't end up flowing through the ATP synthase. And when they flow through freely, we don't get that phosphorylation reaction occurring. We just get that exothermic reaction of the oxygen being oxidized and being converted into water. And so we get this energy production that doesn't get used to fuel this endothermic reaction. It just produces energy in the form of heat. All right, so uncoupling of the electron transport chain results in heat. It results in heat production. The energy, let's see if I can graph this a little. All right, so this is our uncoupler right here, and it's allowing the hydrogen to move in through a different spot in the membrane, not ATP synthase. And so we're not getting this, we're not fueling this endothermic reaction here, but we are still doing this very exothermic reaction oxidation reaction of the hydrogen ions plus the oxygen that will create water and heat, energy that is not going to be used now to fuel our phosphorylation reaction. It will just be given off as heat. All right, so that's what this slide is saying, that if we uncouple those two reactions, then the ATP is not made, we just make heat. We release that energy as heat instead. And there are some tissues that actually have uncouplers on purpose for the purpose of generating heat. There's a type of fat tissue called brown fat that contains uncouplers that they generate heat. And this is important in a lot of animals that hibernate. I've got polar bears pictured here, need to stay warm in the winter. And also in babies. Babies have a fair amount of brown fat that goes away with age, and that's because their little brains, their little hypothalamus, which is supposed to regulate temperature, isn't quite mature enough yet and ready to do that. And so they have the specialized fat tissue that helps to keep them warm. But babies are sort of notorious for not doing a great job of self-regulating their body temperature as well as adults, which is why you have to keep them nice and bundled and warm and safe from the elements. Or likewise, when it's hot out, make sure that you declothe them so that they can don't get overheated. So uncouplers can also be consumed in drug form and chemical form. So there is a supplement called 2,4-dinitrophenol, or DNP for short, and it was popular in the 1930s as a diet aid, and it is still around. You can still get it at like nutrition supplement stores. It's still used by a lot of bodybuilders, but it is dangerous because what it does is it uncouples those reactions of oxidation and phosphorylation to make ATP. And so instead of making more energy, your cells make more heat. Now remember, when you build up ATP, ATP will shut down glycolysis, so you stop breaking down glucose and you send it to storage, either as glycogen or as fat. But if you aren't making more ATP, instead you're generating heat, you just keep breaking down glucose. So it does increase your calorie burn in the sense of you are breaking down or metabolizing more glucose. And instead of generating ATP, though, you're generating heat. And this can actually raise your body temperature to dangerous levels and cause hyperthermia. And people have died from doing this. So do be very careful. I don't recommend taking this as a weight loss supplement or a diet aid, but there are people that have and have suffered really negative health consequences as a result, but that's the science of how it works. Alrighty, so now let's do an ATP count again. So we said that NADH and FADHs, they deliver the electrons that fuel, that electrically energize the electron transport chain. So NADHs result in roughly 10 protons being pumped, and FADH2s result in roughly 6 protons being pumped. All right, the reason that NADH produces more protons than FADH2 is in this slide back here. Okay, notice NADH delivers electrons at pump 1, and so there's protons pumped here, here, and here, versus FADH2, which delivers them at complex 2. I think I misstated earlier in the lecture. I said complex 3, but they drop them off at complex 2. All right, and so they miss complex 1. They don't get to fuel the pumping here. So that's why NADH results in more protons being pumped than FADH2. So anyway, if we count how many ATPs are made for each NADH and for each FADH2, then we can do a much better tally of ATPs. So let's go to glycolysis, that first step, the first pathway. Glycolysis, that 10-step pathway where we're converting glucose to pyruvate. All right, we end up making 2 NADHs and we make 2 ATPs. So those 2 NADHs get converted into ultimately 5 ATPs at the end of the electron transport chain. When we convert pyruvate to acetyl-CoA and we oxidize it in the presence of oxygen, we also make 2 NADHs. So there's another 5 ATPs. And in the citric acid cycle, we make 2 ATPs or GTPs, but we can call them ATPs for all intents and purposes. We also make several NADH and FADH2s that result in several ATPs being formed. So at the end, we have roughly 32 to 38 ATPs. The reason that number varies from textbook to textbook is because these numbers are not exact. So NADH doesn't produce exactly 2.5 ATPs. So that quantification's a little bit rusty, and different textbooks round to different numbers. So that's why you'll a lot of times see 32 to 38 or 36 ATPs generated. All right, a lot of ATPs generated from a glucose molecule. So glucose, we spent a lot of time talking about metabolism of glucose because it is our primary source of fuel, especially for our brains, our neurons, and our red blood cells, and our muscles all depend very heavily on glucose. Some cells can do perfectly fine getting their energy from fat, but glucose is really our primary fuel source. We can store extra glucose, so if our cells have had enough, we've got enough ATP, we don't need to keep doing glycolysis, we can send that glucose off to storage in the form of glycogen, and most of our glycogen is stored in either the liver or the muscles. And if we max out our glycogen stores, we'll actually turn the glucose into glycerol and ultimately convert it into fat. So that's something we don't usually want to do. When our glucose levels are low, like in between meals, we can break down the glycogen into glucose, and that's called glycogenolysis. And that is the process of breaking down glycogen into free glucose molecules when our blood sugar is low. If our blood sugar is really low and we really need some glucose and we don't have any, we can make it from other components of the cells, namely proteins. We can turn proteins into glucose, and that's a process called gluconeogenesis. So when people are on very low carb diets, that are high in protein and high in fat, this is what their bodies tend to do. They tend to convert that protein into glucose as needed. But that being said, we can also generate energy from fats and proteins as well. We can generate ATP from them. So when we do this from fats, the process of converting, we do it in a similar way. We convert our fatty acids into acetyl-CoA. Remember, acetyl-CoA then just gets fed into the citric acid cycle and then into the electron transport chain. So the process of converting a fatty acid into acetyl-CoA is called beta oxidation. So just like we had glycolysis converts glucose into pyruvate and ultimately acetyl-CoA, beta oxidation is like the glycolysis of fats, is how I like to think of it. It's only 4 steps, and a fatty acid has to go through it repeatedly. So a glucose molecule goes through 1 round of glycolysis to get broken up into 2 pyruvate molecules, but a single fatty acid that has 18 carbons is actually going to go through 9 rounds of beta oxidation to make 9 acetyl-CoA molecules. So that's why fatty acids are worth so much energy. When you look at a nutrition facts label, you'll see that carbs they provide 4 calories per gram, 4 kilocalories per gram, and fats provide 9 calories per gram, twice as much. And this is why fats are very high energy. They contain a lot of energy because we can make a lot of acetyl-CoA from them. So that's why fats are more energy dense, is another way to say that. They're more dense with energy. So one cycle, if we compare one cycle of beta oxidation to one cycle of glycolysis though, Glycolysis produces more per cycle. So one glucose molecule going through one round of glycolysis produces 4 NADHs, 2 ATPs, and 2 acetyl-CoAs. Beta oxidation of a fatty acid, one round, only produces 1 NADH, 1 FADH2, and 1 acetyl-CoA. So even though we can get more energy from fats, it takes longer because we have to do 9 cycles of beta oxidation to get all that energy out. So that's why we use glucose primarily for energy, because it's more efficient. We can get more out of one round of glycolysis quickly than we can out of beta oxidation. So if we were to completely digest and metabolize that 18 carbon fatty acid, we would ultimately generate 122 ATPs compared to the 32 that we got from one glucose molecule. It would just take longer. It's sort of a slower process. So fats are often used as an energy source when we are at rest, when we're not exercising, when we don't need energy quickly, when we can just need sustained maintenance levels of energy, and our bodies can do it at a more, slower process. So when you're sitting, or you're walking, or you're doing really low impact activities, you're burning more calories of fat than you are carbohydrate, versus when you're exercising and your body's really depending on glucose, strongly for glucose. Alrighty, so if we break down, when we break down our fatty acids, one of the things that if we have a buildup of acetyl-CoA in the liver, it can lead to the production of ketones. And so the process of metabolizing fats is often referred to as ketosis, because it does generate ketones. Too many ketones, an excessive amount of ketones, can lead to acidification of the blood, which is bad. This very rarely happens in healthy people. Even if you were on a super, super low carb diet, you were eating lots of fat and like 0 carbs, the idea is to stimulate ketosis, breakdown of fat, but a healthy person would not get to a dangerous level of ketosis. Where it becomes dangerous is in people who have diabetes. In diabetics, they are breaking down fat at a really accelerated rate because their cells cannot cannot take up glucose, and so they can get to a point where their blood becomes acidified. So ketoacidosis is something that is a potential dangerous side effect of uncontrolled diabetes. And then lastly, energy from our proteins, from amino acids. So we already talked about one way that we can get energy from proteins is by converting them into glucose in a process called gluconeogenesis. But another way that, so we can do gluconeogenesis of some of our amino acids and produce glucose, which then can be converted to pyruvate and acetyl-CoA, and then acetyl-CoA goes through the citric acid cycle. Of the 20 different amino acids we have, though, they each can, all of them can be converted into some component of the citric acid cycle. So they can either be converted into an intermediate component and enter the citric acid cycle that way. They can be converted to acetyl-CoA. They can be converted to a precursor to acetyl-CoA, or they can be converted directly to pyruvate. So there's lots of different ways. You don't need to know how each of these amino acids is metabolized. Just know that the way we get energy from amino acids is by converting them into one of these substances, either the starting material, acetyl-CoA, or an intermediate material in the citric acid cycle. Did I want to say anything? Oh, our proteins rarely are used for energy. We tend to use amino acids for building proteins. We have so many necessary functions of proteins that energy is really they're a last resort use of energy. So we really only start using amino acids for energy when we are starving, or if you're on like a super low carb diet. So only about 10% of our energy really comes from amino acids. We excrete extra amino acids. So we store extra glucose as glycogen or as fat. We store extra triglycerides as fat, as triglycerides in our fat tissue. But extra amino acids get broken down into urea and excreted in the urine. So we don't tend to store extra amino acids. And that's, what I wanted to say about that. All right, so this slide, I just want to map out everything we've just talked about. And this is important that you can do this. You only really understand this material if you can draw a map of it yourself. So when we talk about mapping it, what we're talking about, this process of catabolism, okay? We have our 3 macronutrients, carbs. So you're eating a burrito, all right? And there's carbs, there's starch in the tortilla, and there's lactose in the cheese, and there's fiber that we're not going to break down in the beans. All right, there's fat in the meat and in the cheese. And there are proteins as well in the meat and the beans and the cheese. All right, so carbs are polysaccharides, right? They are long chains of monosaccharides. So that's what we're going to break them up into. We're breaking up our carbs into monosaccharides. Monosaccharides. The 3 monosaccharides are glucose, fructose, and galactose. Right? And we do metabolize them similarly, but of course the one that's most abundant is glucose. So I'm going to write glucose here. Glucose is the most abundant monosaccharide, but it's not the only one. Our fats can be broken down into their component parts, glycerol or a monoglyceride, and free fatty acids and fatty acids. So remember, fats are the one biomolecule that are not polymers. They're not made up of long chains of repeating units. They're made up of 1 glycerol and 3 fatty acids. And then proteins are polymers. They are made up of building blocks called amino acids. And so we ultimately digest proteins into amino acids. Okay, so now that they are, so this takes place, the large biomolecules, the carbs and fats and proteins, they are in our intestines. And the enzymes, intestines, all right, and then we have enzymes in the intestines like amylase to break down carbs, and lipases to break down fats, and proteases to break down proteins, that now these simple building blocks are small enough to get absorbed into the bloodstream. So this is what we can absorb into our blood, and our blood carries it around to our cells, and then our cells can take in the glucose and convert the glucose into pyruvate. Pyruvate. I don't know if you can hear the TV downstairs. My husband's watching it on like movie theater volume. All right, so this process here, this arrow, we could label it as glycolysis. This process of converting glucose to pyruvate is glycolysis. All right, under aerobic conditions, in the presence of oxygen, You could write aerobic. I'm just going to write O2. All right, when oxygen's around, then pyruvate is converted into acetyl-CoA. And acetyl-CoA, all right, acetyl-CoA can be converted into fatty acids if you have a lot of it. If you have more than you need, you can convert it into fatty acids. Fatty acids can also be converted into acetyl-CoA. So this is sort of a two-way street. This is a reversible reaction here. Acetyl-CoA can get fed into the citric acid cycle. All right, so acetyl-CoA is in the citric acid cycle. The citric acid cycle is 8 steps. And some of the important byproducts of the citric acid cycle are carbon dioxide, which we exhale out because it acidifies our blood, and also those electron carriers, NADH, and FADH2. And these guys, remember, they're electron carriers. They literally pick up electrons from redox reactions that occur in the citric acid cycle and carry them to the last part of this whole shebang, which is, so these two really are what are going to the electron transport chain, which, if this is my phospholipid bilayer, which I'm too lazy to actually try to sketch out, all right, there are 5 protein complexes. 1, 2, 3, 4, and 5. All right, they drop off electrons, so NAD or NADH drops off electrons at complex 1, and FADH2 drops off electrons at complex 2. And these electrons flow through complexes 1 through 4. All right, and they generate electricity, and the electricity is used to pump protons into this intermembrane space. Complexes 1, 3, and 4 pump protons or hydrogen ions into this space up here, and they build up a concentration gradient, and they really want to come back in, and they ultimately do come back in through complex 5, our ATP synthase. All right, and so those hydrogen ions and those electrons that are coming out at the end of the electron transport chain couple up, or they cuddle up to water, or sorry, to oxygen to form water. And this is a very exothermic reaction. It generates a lot of heat, all right, or a lot of energy, I should say. So I'm going to say plus energy. All right, and the energy from this reaction ends up getting coupled with this reaction here, ADP, plus free phosphate. That's a P, to form ATP. This is a very endothermic reaction, so it requires energy. So the energy from this oxidation reaction of oxygen and hydrogen and electrons becoming water ends up allowing us to generate ATP. It provides the energy that's needed to make ATP, and then ATP can get hydrolyzed, and that is an exothermic reaction that fuels other reactions in the cell. So this process of converting fatty acids to acetyl-CoA here, I'm going to label this, is called beta oxidation. Beta oxidation. I'm just going to do beta ox. All right. And amino acids can also be converted into acetyl-CoA, or they could be converted into different components of the citric acid cycle. I'm going to just label this TCA. It could also be called the Krebs cycle or the citric acid cycle. And down here, what I've drawn is the electron transport chain, which is often abbreviated ETC. All right, so this is really what I've mapped out here primarily is catabolism. Catabolism, all right? Catabolism is all of these reactions that go sort of down, the down arrows. But we also have anabolic reactions. I don't know how to change my pen color in this tool, okay? But if I could, I would draw a different color of reactions where we are synthesizing things. So synthesizing amino acids, synthesizing proteins, all right? Synthesizing carbohydrates, complex carbohydrates, polysaccharides from monosaccharides. Those would be anabolic reactions. And those are reactions really that were the topic of chapter 6. Okay, that I can't even draw anymore. That were the topics in chapter 6. So when we talk about linking monosaccharides together to make glycogen or amylose or cellulose in plants, all right, those would be anabolic reactions. So that is the end of chapter 12. I know metabolism is really heavy. It's a lot of detail. So you may need to rewatch this or watch some supplemental videos to help get this whole picture to come together. But it is really important in anatomy and in future biology classes to understand how cells make this very important molecule here of ATP from macromolecules in the diet, fats and proteins, but primarily from carbohydrates, our main carbohydrate being glucose. The end.